Hello, my beautiful friends and castmates. I promise that is the end of my bee humor. My name is Victoria Melanson, and I will be presenting the facultatively eusocial sweat bee, M. genalis. For anyone who wants to read along, I will be posting more or less what I say below. In this presentation, I'm going to go over the development of my chosen eusocial insect, Megalopta genalis, starting with the food, the brood, caste determination, nest formation, backup nest strategy, and finishing with the potential evolutionary developments that have led to this unique creature. As a little background information to set the scene, Megalopta genalis, or M. genalis, as I will switch back and forth between, is in the order Hymenoptera, as you all know, is bees and wasps. But they're in the class Halictidae, and I'm pointing this out because these are the sweat bees, which are attracted to perspiration. This is a neotropical bee that is native to Central and South America and forages crepuscularly at dawn and dusk. They can live in hollowed out sticks with 1 to 10 females per nest, typically, but can also live in nests of 2. Being facultatively eusocial here means that for them to be eusocial, there's a caste-based society in place that creates a separation of the bees into reproductive castes, so queens and workers. In this species, caste determination is considered to occur when they are already adults and often determined by aggression from, for the most part, larger queen. And facultative expression of a social behavioral phenotype means that a particular physical phenotype has to be continuous and overlapping. In this case, it would be size. So, and here we can see where the bees are from. So this is the Barrow, Colorado island that one of the studies I'm citing gathered specimens from. And the overall theme going forward is going to be the development of this bee and how the different developments contribute to it being social. So the brood. I would like to start the brood section with a disclaimer that not much is known about the mating history of M. genalis, so we're going to gloss over it. The young, however, as an egg, are placed on top of a pollen nectar dough ball and then closed off to the world. This is a crucial time for the young, as it is during this development that body size is determined. As you can see here, the egg as it develops. After emerging and being a child bee for a bit, M. genalis helpers then go off to forage at the tender age of 1 week old. So we're going to go deep into the brood brain. After measuring volume of brain areas in social reproductives and workers, solitary reproductives, 1-day-old bees, it was found that social reproductives showed increased development of the mushroom body compared to social workers and solitary reproductives. The mushroom body, MB, in question, right here, is a portion of the brain that is suggested to indicate evolved developmental patterns, and individual life experience through its morphology, so as it changes. When an individual of the species switches environments from confined nest to complex sensory stimuli and navigational requirements, the MB changes morphology, changes shape. Because M. genalis is able to live in the smallest possible social group, but still exhibits similar MB morphology as species, they have large colonies. Therefore, so they have the same MB morphology as species that have large colonies, and therefore it is suggested that dominance, something highly exhibited in this species, is more likely to be the cause that shapes brain size and development instead of previously assumed amount of behavioral interactions and contact rates. Sorry, I had to regather my thoughts there. So the food. Trophallaxis. Trophallaxis is the exchange of liquid food among adults. This is important because that means that you have an adult feeding another adult. Trophallaxis is debated as to whether or not this is ritualized or simply a typical dominant submissive behavior. As shown by Wcislo, it could be a ritualized social behavior because under experimentation, trophallaxis still occurred even though no physical aggression was possible. It is shown that trophallaxis occurs more frequently at the entrance of the nest when the primary residents would wait, and it happened most frequently when the queen became agitated and increased aggressive behaviors and tendencies until trophallaxis was performed. I can relate to that. Additionally, survivorship of females and males increased by 50% under experimental conditions. So M. genalis is a mass provisioning species. This means that they put away all of their food that could be used or needed for the next round of bee larvae, the brood, before they are laid into their cells. And other than that, they do not provide additional care for their brood other than to protect the nest. Here we have an example of an insurance benefit, or otherwise known as assured fitness return, of reducing adult larval interactions to only that of the queen and the brood. In this fashion, the queen can directly manipulate the cells if she so chooses in a way that benefits the colony and herself. So for M. genalis, quality over quantity matters greatly. Nutritional inputs for individual offspring can have a potentially huge social implication. Higher quality pollen masses are more often provided to females over male eggs in social nests, which makes the females harder to provision for. This manipulation allows for maternal effects to dictate whether her offspring will be male or female, and whether a female will be a gyne, a reproducer, or a worker. While body size does not always correlate to fecundity, which is their ability to reproduce, it does indicate whether or not the individual would win a dominance interaction or show of force. More food. Many nests in one area are often found at the same location and produced offspring throughout the tropical dry and early wet season at the same time. It's a lot of bees in one area all at once. It is imperative that the foundress of a nest balance the line of larval nutrition in order to continue her species through either manipulating her own nest and therefore giving lower quality pollen nectar masses to produce more workers, or allowing the opportunity for a gyne to create her own nest, so giving her higher quality nutrition. Or possibly giving this higher quality nutrition could lead to a gyne overthrowing her. To remain a social queen, the foundress would have to provide just enough pollen that her females will remain workers and stay behind to raise the brood, but also be able to reproduce if she were to disappear, but not to give them enough that they would be able to ride the wave of fat, protein, and endocrine stores that go along with that high quality nutrition and allow them to dominate her. In order to forage for pollen and nectar, this is done at dawn and dusk to take advantage of night flowering plants. To achieve this, Megalopta develops relatively large eyes in relation to their body size and can forage at intensities less than the level of starlight. Since larger eyes are metabolically more expensive, it is assumed that vision is very important for Megalopta. Additionally, this bee uses vision and a back and forth hovering technique to learn landmarks around her nest in order to home in upon return. This is interesting because that means that not only do they take advantage of night flowering plants, but they also take advantage of being socially cooperative and getting an advantage over other potential social insects in the area that would not be foraging at this time. And since so many of them are in one spot at one time, this gives them a wider range of food so that they're not competing within their own species. So, caste determination. Now, I don't want you to feel like I buried the lead, but I did purposefully save this for the middle of the presentation by not drawing specific attention to it. This is the meat and potatoes of the species. What makes this bee so special is that it can choose to be solitary or social. The solitary queen is usually smaller and becomes reproductive later, when she will eventually have a singular nestmate who helps raise the brood. The social queen is larger can reproduce sooner and often produces workers first. But these foundresses do not differ in rate of survival, which is very interesting. So we have the 4 female phenotypes: solitary, queen, worker, and replacement queen. The difference in expression of these alternative phenotypes is linked to body size, nutrition, ovary size, hormones, environmental changes, and social conditions. To that effect, we already know that the quantity and quality of nutrition can be maternally manipulated, and therefore the queen can decide on the overall body size and sex of her offspring, and potentially their fat, protein, and hormone stores. It has been cited that queens typically have larger ovaries, higher juvenile gonadotropin, and significantly higher vitellogenin tetras than her workers. In terms of environmental and social conditions, if a queen were to disappear or die or is dominated, a reproductive female may ascend. If one of the workers is able to reproduce, in the absence of the previous dominating female, she may enlarge her ovaries in order to produce a brood and retain her new dominance. Since it is the previous queen that forces the female to have reduced ovaries, despite exhibiting high levels of behavioral plasticity, it is the social demands of the queen that enforces a physiological difference on her workers to create their reproductive behavior. And therefore it is this combination that may decide whether or not an M. genalis foundress would create a social or solitary colony. The nest. Within a single population, a foundress may be social or solitary, as we just discussed. Social nests, so defined as being a colony of 3 or more, have a strong division of labor, with the workers doing the foraging, some brood care, and feeding the queen and other nestmates occasionally through trophallaxis. Nests are founded by a solitary mated female with a 25 to 30% co-founded rate in tree branches or vines at the start of the dry season in mid-December. She forms lateral brood cells, and from there it is unknown to date what kind of mating biology occurs, as we mentioned earlier. Nests can last for up to 10 months, but 50 to 60% of all nests fail within 5 weeks of being founded. This is why it's so important that some of them can create their own broods so that to overcome that massive fail rate. M. genalis has an incredible social plasticity, as aforementioned, and this is exemplified in a study by Kapheim et al., who discovered that some nests contain interspecific nest parasitic females and some nests that neither follow a typical solitary or social nest pattern. In order to be a successful solitary nest, the foundress will typically produce males first, and following female broods will disperse. In contrast, social nests are more likely to produce females first, with some being gynes and some as non-reproductive workers. Incredibly, since all nests are founded initially by a single foundress or co-founded, for the first brood, the main reproductive female must forage and supply her offspring with food. Therefore, all bees, including queens, have experienced foraging at some point in their development. Home sweet home. Another remarkable ability of this bee is that it seems to be able to sense when something is awry within a brood cell, like a psychic ability. During an experiment performed by Nonacs et al. In 2015, a foundress will actively respond to a drastic change within a cell, despite the fact that it is sealed. Physical barrier. Changes that can trigger her include unauthorized nutritional change to a larvae, and this will cause her to open the cell within days. And the death of a larvae can trigger her to open it immediately. This has incredible social implications because it pairs nicely with the assumption that the state at which a larvae is controlled is so very crucial to the social structure that the foundress hopes to create that she will keep the adjacent cells clean if one dies and open and manipulate any that are getting too much or too little food. So now we're going to back up that nest. So backing up a nest leads to a theory that head size is possibly more important for females than males, because under a predictable order of operations, a larger head would mean larger mandible and adductor muscles. Having larger mandibles and adductor muscles would definitely be useful for making a nest and excavating brood cells. Replacement queens are able to reproduce just as well as the previous queen after enlarging her ovaries and asserting her new position among colony mates. The fitness outcome of a foundress being able to exhibit polyphenism in choosing to be social or solitary lies within producing males first as a solitary queen. since males are less costly to provision because they don't need that varying difference of quality of food, it is easier to produce a direct reproductive potential with little investment. The risk of being social is that nearly 10% of social queens fail to retain workers. However, social nesting and having a larger colony is heavily favored for daughters in social continuity, because then, if the queen dies or disappears, her daughters are saved from the risk of starting their own nest and can simply take over from her mother. This enforces the evolution of worker behavior, although this benefit is outlived if the substrate that the nest is made in is already at an advanced state of decay. Now I would like to wrap up my presentation and the development of the bee with the evolutionary development of Megalopta genalis. The ability to have an active parental brood care is hypothesized to be a pre-adaptation to eusociality, and it was previously assumed that a closed cell creating that physical barrier precludes caregiving. One of the proposed mechanisms of parental care leading to eusociality is the hygienic hypothesis, which states that since they live in such close quarters, that to maintain healthy social societies, being proactive against potential health issues like pathogens created by disarray and disuse can be mitigated by opening a brood cell and removing feces or a dead or non-viable larvae. And this would increase brood health and therefore reducing costs associated with social life and increase long-term productivity. Abdominal caste-specific differences in gene expression were found within M. genalis, which is very cool. Additionally, it was found that caste-specific differences in gene expression was found to overlap with other eusocial bee species. With this overlap and unique phenotypic plasticity, the development of genetic variation within M. genalis can serve as a proxy for ancestral variation and how they may have evolved to translate into a selection for permanent reproductive and non-reproductive castes. This development of plasticity of workers biased gene expression is consistent with the hypothesis of gene accommodation, and this ancestral plasticity in the ancestors of obligately eusocial species of bees is what could have led to the evolution of the different castes and caste determination system described thus far. On the subject of genetic accommodation, let's take a closer look at that. Genetic accommodation is when novel phenotypes, or just different phenotypes, can form from a single genotype solely based on a changing conditions during development, yielding a special form of developmental plasticity that allows for several unique species to derive from a recent common ancestor. Here we have M. genalis expressing gene accommodation, as we said, by forming the complex phenotype of caste-determined eusociality from ancestral gene plasticity. This complex phenotype represents major innovation on the part of the bee. It creates the extreme differences we talked about in morphology, physiology, and behavioral, social, or solitary. These differences arise in part due to the changing environmental conditions and genetic variation through development. Similar research of other social insects, such as ants, wasps, and bees, have shown that they, too, exhibit hundreds of interacting ancient and pleiotropic genes that contribute to social behavior. Kapheim et al. Suggests that this indicates that genes regulating social behavior in eusocial species descended from genes that had an entirely different function in their solitary ancestors, and further posits that this change could have been raised from regulatory change to allow these genes to be expressed in new spatiotemporal and epistatic contexts. And therefore, social traits may emerge not only from the environmental changes that we've discussed throughout this presentation, or the demand that comes with it, but also from evolutionary changes in the way ancient genes are expressed. Thank you for watching and listening to my presentation.